Hello everyone, welcome to the channel Microbial World. Today we will be discussing about acid-fast staining. The reagents used in the staining process, principle, procedure of staining and interpretation of results will be discussed. Acid-fast staining is a differential staining technique which was first developed by Ziehl and later on modified by Neelsen. So this method is also called Ziehl Neelsen staining technique. Acid-fast stain is a differential stain that is used to identify acid-fast organisms such as members of the genus Mycobacterium. Acid-fast microorganisms are characterized by wax-like, nearly impermeable cell wall. They contain a lipid, mycolic acid, and large amount of fatty acids, waxes and complex lipids. This type of cell wall is resistant to most compounds. Therefore, acid-fast microorganisms require a special staining technique. The ability of the bacteria to resist decolorization with acid alcohol confers acid fastness to the bacterium. The main aim of this staining is to differentiate bacteria into acid-fast group and non-acid-fast groups. Reagents used in acid-fast staining. Primary stain is carbolfuchsin. Decolorization solution used is acid alcohol decolorizer. Counter stain or secondary stain used is methylene blue. Principle. When the smear is stained with primary stain carbolfuchsin, it solubilizes the lipoidal material present in the mycobacterial cell wall. And by the application of heat, carbolfuchsin further penetrates through lipoidal wall and enters into cytoplasm. Then all the cells appears red. Then the smear is decolorized with decolorizing agent 3% HCl in 95% alcohol. But as we said before, acid-fast microorganisms are resistant to decolorization with acid alcohol. And that is due to the presence of large amount of lipoidal material in their cell wall, which prevents the penetration of decolorizing solution. The non-acid-fast organisms lack the lipoidal material in their cell wall, due to which they are easily decolorized, leaving the cells colorless. Then the smear is stained with counter stain methylene blue. Only decolorized cells absorb the counter stain and take its color and appears blue, while acid-fast cells retain the red color. Procedure of acid-fast staining. Prepare bacterial smear on clean and grease-free slide using sterile technique. Allow smear to air dry and then heat fix. Cover the smear with primary stain carbolfuchsin. Heat the stain until vapor just begins to rise, that is about 60 degrees Celsius. Do not overheat. Allow the heated stain to remain on the slide for 5 minutes. Wash off the stain with clean water. Cover the smear with 3% acid alcohol for 5 minutes or until the smear is sufficiently decolorized, that is pale pink in color. Wash well with clean water. Cover the smear with secondary stain methylene blue for 1 to 2 minutes using the longer time when the smear is thin. Wash off the stain with clean water. Wipe the back of the slide clean and place it in a draining rack for the smear to air dry. Do not blot dry. Examine the smear microscopically using the 100x oil immersion objective. Interpretation of results. Acid-fast cells appear bright red to intensive purple or red color. Straight or slightly curved rods occurring singly or in small groups may appear beaded. Non-acid-fast cells appear blue in color. So this is all about acid-fast staining. Thank you.